Today we have Professor Rainer Bischoff with us. Good morning, sir. Professor Rainer Bischoff has obtained his PhD in chemistry from the University of Göttingen, Germany. He continued his professional career as postdoc at Purdue University and joined the biotechnology industry in 1987 as scientist at the Transgene SA in France, where he was a leader of a preclinical research project concerning the use of alpha-1 antitrypsin variants in the treatment and prevention of septic shock, and continued to bringing one of the first therapeutic proteins developed in Europe to the market. He joined AstraZeneca in 1998 as leader of the protein chemistry team and later as head of the target development section. He started his present position as head of analytical biochemistry department at the University of Groningen in the Netherlands in 2001. His research group has collaborations with various medical research groups in the areas of disease biomarkers and therapeutic proteins, with chemistry groups in the areas of targeted protease inhibitors, and with informatics groups concerning data processing and statistical data analysis. With this brief introduction, I welcome you again, sir, and you may please begin with the lecture.
Yes, thank you very much, first of all, for the invitation to give a workshop. It's amazing with technology these days. You can give a workshop half around the world, sitting in your own office at home, due to Corona restrictions, obviously. But it's very nice to be back in India. Let's say I've been in India a couple of times, but unfortunately this time only virtually. So I will share my screen and hope that everything will work out. And then I can start. Can you see my screen?
Yes, sir, it's visible.
Okay, great. So my talk today will be pretty much about the bioanalysis of proteins, and that's maybe a little different from what you might expect, but I will link it to post-translational modifications and also to modifications in general on proteins. And what I would like to allude to is pretty much the modification that proteins undergo also in vivo. So we probably heard a lot about protein characterization, the characterization of therapeutic proteins, and this is obviously a very hot topic, and it's done in very great detail. But to my surprise and to surprise of others, there's very little analysis done once the proteins are administered to a patient. And this so-called biotransformation is one of our topics, and I would like to link that also to the technology and the methodologies that are needed to do that. You see below is our research institute. It's called the European Research Institute on Aging. So it's all related around aging. So not really a therapeutic protein production site, but therapeutic proteins are one of our topics, and we work in a European research project on that. Let me see. Oops. Oh yeah, well, it takes a moment to move. So just because, you know, India is far away, I thought I'd give a one-slide introduction of where I'm working, and that's in Groningen. It's a city in the northeast of the Netherlands. The Netherlands is not so big, as you know. Everybody knows probably, or at least has heard about Amsterdam, the capital of the Netherlands, which is down here. And Groningen is about 200 kilometers northeast in the Netherlands. It has the second oldest university, which was founded in 1614. So Groningen is really a university town with a lot of students. I think a quarter of the population is actually related to students or the university. Where I'm actually working, although I'm at the Faculty of Science and Engineering, I'm working at the University Medical Center, which is this part here. So this is where, you know, it's a hospital essentially, it's patient care. And here is our preclinical area where we have the research labs. Actually, our institute is not even on that slide yet, since it's pretty new. So this is the preclinical research, and I show this because I like this very much, because we have a very close relation to the hospital, and that's obviously very important if you want to look at biotransformation, but also at biomarkers, as was mentioned. So all kinds of clinical research projects, important to have the clinical research close by, so you can essentially just walk over and you have an easy way of collaborating, although I'm a chemist by training. Okay, with that brief introduction, let's make an overview of what I'm going to talk about today. First of all, the protein bioanalysis methodology, And my statement is one size does not fit all, and I would like to introduce you to different bioanalytical assays and the sometimes a little bit nerve-wracking result that not every assay gives the same result, and why that could be. Then the quality attributes and biotransformation. I'll focus on Trastuzumab, well-known therapeutic protein, which we work on also in the European Union project. And the quality attributes and what is critical, what is not critical, and what happens in vivo to Trastuzumab in this case, as far as we know, because we're still working on that. Then another topic that we've worked on is anti-drug antibodies on the protein alpha-glucosidase, which is used as an enzyme replacement, a therapeutic protein in Pompe disease. And so it is a lifelong treatment for starting with childhood. And thus anti-drug antibodies can be a very severe impediment to the treatment of patients. It's very important to determine that and also to try to prevent or to block anti-drug antibodies, which is not really our topic, but first you have to see who is developing these anti-drug antibodies. And I would like to introduce that you can do this very nicely with LCMS actually, it's a mass spectrometry workshop also. And at the end, allude briefly to the possibilities or also the challenges of high resolution mass spectrometry in this area. So let's start with proteins. Proteins, whether they are therapeutic or non-therapeutic, they are all, it's the same. They'd say they're made of the 20 amino acids with post-translational modifications. And here we see some of the changes. We call them proteoforms. And this already alludes to the reason why bioanalytical assays don't always give the same results, because you can have, for example, here a methionine oxidation, you can have a deamidation on a protein, like here you can see that. You can have a proteolysis, which happens obviously in vivo. It's not very studied in great detail, but I think with LCMS assays, we have now the unique opportunity to also study these transformations in complex mixtures. Here's the oxidation symbolized, but also we can have just changes in 3D structure, which may actually affect the interaction with a detecting or capturing antibody in a bioanalytical assay, and then the protein will no longer be detected while it may still be around and while it may still be active. So it's important to keep in mind that the result with a bioanalytical assay, be it a ligand binding assay or an LCMS assay or another assay, is never gives you the complete answer. It just tells you about what it actually measures. Another reason for varying results in these assays is competition for binding. Many protein assays in bioanalysis use a binding partner, be it an antibody, an Elisa, or another affinity binder. And these can be competed with. We often refer to proteins as if they were always in free form. They would circulate by themselves, but this is usually never the case for any protein. They're always interacting with something. So a monoclonal antibody, for example, is engineered to interact with a target protein. And so this target protein may actually block the binding site. Usually it's a receptor, but these receptors can be cleaved off the membrane of cells and they can circulate the extracellular domains. And they may, for example, interfere with the binding of the detecting or the capturing antibody. Proteins can dimerize and many other things can happen. And at least last not least, they can be bound to anti-drug antibodies, which would also potentially affect the result of the assays. The problem is that we are looking through a keyhole. We never look at the whole protein with our assays in bioanalysis. We usually use a recognition element that is only a small part. In a ligand binding assay in LBA, this will be an epitope on the protein, which is recognized by the antibody. So if this epitope is modified, the interaction may not be there anymore. And if the protein is modified somewhere else here, I hope you can see my cursor, I should ask. Is that okay? Maybe some quick answer. Can you see my cursor? Anybody can answer?
Yes, sir, we can see. We can see.
Oh, great. So,
oh,
excellent. So then, then you won't see it in your assay. Well, with LC-MS, we often just cut the protein up into bits and pieces, so-called signature peptides or proteotypic peptides, and we look at 1 or 2 or 3, but for a normal protein, a big protein, there may be 50 peptides generated in a tryptic digest. And, if this one is modified, we will see it, but if anything else is happening in the protein, we don't know it. And we also lose the connectivity. So if we see a modification in one peptide, another peptide, we actually don't know whether this belongs to the same proteoform. And that's actually also a critical question for protein product characterization, not only for bioanalysis. Here's an example from a review by Hall, which I think is quite nice and also quite interesting to read. There's not so much about the biological fate of proteins. Here it says chemicals, it's the name of the journal, but here we talk about the biological fate of proteins. Here's an example where you have an Fc, an antibody, conjugated to another molecule. I think it's an anti-diabetic peptide. And when you measure the pharmacokinetics of such a molecule, which is usually done by a ligand binding assay, then you may have different pharmacokinetics depending on where the molecule binds. Either you measure the whole thing, with the conjugated peptide attached, or you just measure the antibody, and they have different elimination rates. And so you can get different pharmacokinetics depending on what kind of assay you use. This can be confusing, but it's often also before LC-MS, it was never recognized. You just get a result, but you actually don't know, what you really measure in your biological and your bioanalytical assay. This is shown here, an example by Kang et al., where they measure an Fc fragment that is conjugated to the glucagon-like peptide 1 in an anti-diabetic track. So this is really a molecule that is on the market. And there is a cleavage site, and this peptide should be then released and take its action. So this can be used as an advantage. I showed it now until now as a disadvantage, but you can actually look at the release of the peptide. Here you see two different pharmacokinetic curves. The red one is for the total Fc molecule. So that's a signature peptide from that part. And the green one is the intact molecule, which actually goes down as the peptide is released. So that's a signature peptide that covers also the cleavage site. So you can actually see the release kinetics of this peptide from the, let's say, carrier protein. Something that's quite difficult to do with a ligand binding assay, and with mass spectrometry, as you probably know, you can follow many peptides in one LC-MS analysis. So you can look at many different proteins potentially. It's what we do when we do biomarker studies, but also at different parts of one particular protein, which is quite useful for therapeutic proteins. This was modeled here by Furlong, but it says essentially the same thing, so I don't need to dwell too long about it. You can have two scenarios. You can have the same pharmacokinetics. If there's no in vivo degradation, they call it here, or cleavage. And if there's a cleavage and you can have two different signature peptides, the red one and the black one, then you will see two different pharmacokinetic curves with time. Recently, we developed an assay with Peter Bülz, who was a PhD student, still is a PhD student, almost finished in the group. And we measured the pharmacokinetics of recombinant human growth hormone in red plasma together with a company in Denmark called Ferring Pharmaceuticals. And what we actually see, it shows three different peptides. I can show the sequence here. So one is in the middle, so this SNL peptide. One is at the N terminus, and one is more towards the C terminus. And you can see that while the actual pharmacokinetic curves are very similar, the absolute concentration values are quite different depending on where the peptide is located in the protein. We don't know for sure, but it's likely that the peptides that are more on the outside of the protein, towards the N or C terminus here, show lower concentration. So there's probably, in vivo degradation of this protein, which will slowly but surely, eat into this area, so that the peptide that's right in the middle is, let's say survives longer, you could say, in vivo than the ones that are closer to the edge. So while it makes maybe the results a little bit more complicated, you don't just get one number. If you can interpret the results of LC-MS, you can actually get a lot more insight of what is actually happening. The second part of this first part, let's say, is that the immunocapture, the capture agent in general, plays an important role. Many assays, also LC-MS assays, if you go down to low concentration, you need to enrich your protein. And often that is done with monoclonal and sometimes also with monoclonal antibodies. So here in this paper by Fischer et al., you can see that this can have a significant effect. That's actually a very interesting effect. Here you have the monoclonal antibody or the therapeutic protein or Trastuzumab. And it's either captured with a monoclonal antibody or a polyclonal antibody. And you see, well, if you do this at the beginning of your pharmacokinetic curve in serum, then there's no difference between the two. But as the time goes on, your result, and this is a logarithmic scale, with the monoclonal antibody are, a factor 5 at least lower than with the polyclonal antibody. So these are not small differences. So this could mean that either the epitope is modified of the monoclonal antibody, and since the polyclonal antibodies, I should say, have multiple epitopes, they still work. But in this case, what they found is that the target molecule CD20 actually binds close to the epitope of this monoclonal antibody. And as you know, as it interacts with this molecule, then the response in this ligand binding assay gets lower and lower. So that's quite interesting that you see these differences. And this also explains sometimes why it's very hard to standardize assays. We work a lot with clinical chemistry lab, and one of the big problems in clinical chemistry is that comparability of results between different hospitals and beyond the borders of countries and so on internationally is often very difficult because of these kind of interactions, interferences, and also the quality of the immunoreagents. Here's another example. Don't spend too much time on it, but here you see a clear difference between LC-MS and Elisa. And the LC-MS uses an anti-Fc capturing agent, while the Elisa uses an anti-idiotypic capturing agent. So anti-idiotypic means that the antibody binds to the CDR of the therapeutic antibody, while the Fc is obviously the constant part. And you can see huge differences. For example, look at the 10 milligram per kilogram ELISA curve, which is this one here. It's actually below even the 3 milligram per kilogram pharmacokinetic curve for LC-MS. That means there's really a big difference between the two ways of capturing the target molecule. So it's not to say one assay is right and the other one is wrong, but it is very important to understand what's going on. And that's a definitive advantage of mass spectrometry, because you can actually determine what you measure, while in a ligand binding assay, you just get a result. You just get a fluorescence or a chemoluminescence or some result, but you don't really know what the antibody recognized and what was the reason for this result. There it is stated again, what I said. So let's make some intermediate conclusion and then move on to the next part. So be aware that protein bioanalytical assay may be affected by biotransformation, and that may also lead to conflicting results. So something may happen or will happen to the protein. Proteins are not just, they are catabolized and eliminated. So definitely there's degradation going on. They may also be affected by competing events, binding events, circulating target protein, anti-drug antibodies, as we will see. This always applies for assays that are based on ligand binding, on antibody capture or something like that, but also many LC-MS assays, unless they do not use antibodies. Some of them, we are working on that as well, but others too, to avoid antibodies, but that's not possible if you want to go to really to the low nanogram per milliliter and below concentration in a complex matrix. For now, maybe it will be in the future. And the immunocapture reagents may affect the results, as shown on the example of polyclonal versus monoclonal antibodies, or target-specific versus more generic Fc binders in this case. Okay, so moving on to the second part. So there is, as introduced already, a lot of heterogeneity in proteins. And when I worked on recombinant proteins already in the 1980s and early 1990s, this was a new concept to regulatory agencies. FDA and others were not used to getting, to having drug molecules that are not homogeneous. They were used to chemically synthesized molecules, and you had a one structure, and this was clear, and there was an NMR spectrum, and everything was clear. But for therapeutic proteins, they had to get used to modifications. So here is a table from Alain Beck, who published a lot and nice papers in this field on therapeutic antibodies. And you can see there's a whole range of modifications, which you've heard about already, from charge modifications to oxidations to glyco modifications or variants. And here I compared what do you actually see in natural antibodies, because that's in a way what we're trying to mimic, and what do we see in recombinant antibodies, a little bit with the idea, well, what is now modifications, yes, but what's important, what's critical, what are the critical quality attributes versus just changes that happen. For example, you see the pyroglutamic acid at the N-terminus, that's almost always present in natural antibodies, probably protects them against degradation. While in recombinant, this is varied. But if you find a pyroglutamic acid, that's not really, you could say, a problem, because it is also there naturally. So that's, that happens. Also, asparagine deamidation and aspartic acid isomerization, which I will allude to a little bit later, they are quite common. But sometimes they can affect the activity. Sometimes they can affect also the pharmacokinetics. For example, the methionine oxidation, some methionines are critical, like 258, which then leads to a much shorter half-life because they do not interact with the neonatal FC receptor anymore. That's low in natural antibodies, but it can be quite substantial in recombinant antibodies, especially also due to the handling and the storage of the antibody, and so on and so forth. So we all had to get used to defining the heterogeneity of these products and then trying to keep them constant. Sorry. Now, this concept has now been accepted by the regulatory agencies, but still you need to keep these quality attributes within certain margins. Let's move on to Trastuzumab as our example. So as an introduction, and most of you may have heard about it, Trastuzumab binds to the so-called HER2 receptor. The HER2 receptor is a member of the epidermal growth factor receptor family, which was the HER1 or EGFR, epidermal growth factor receptor, was the first discovered receptor, but later it was found also by homology cloning that there were actually 4 members of this family, and HER2, against which Trastuzumab is directed, is quite a special receptor or special member of this family because it doesn't have a natural ligand, but it can dimerize with all of the other receptors. So it's kind of a promiscuous receptor because upon dimerization, the receptor gets activated as a tyrosine kinase domain intracellularly, and then you can get expression and transcription, cell proliferation, and so on, and also the production of vascular endothelial growth factor for blood vessel growth, which then allows the tumor to grow. So blocking HER2 is a very important event, and Trastuzumab was one of the first therapeutic antibodies on the market, and it's been a great success ever since, because HER2 positive breast cancer mainly, but also other cancers, were resistant to any other treatment until then, and were very aggressive. So the prognosis is very poor for patients with that kind of a tumor. So here we see Trastuzumab and its various levels of interaction. It can prevent, for example, the cleavage, which will also lead to a constitutive activation of this receptor. As shown here, it can prevent this dimerization by blocking a domain, so that also interferes with activation. It also attracts immune cells, so natural killer cells, to the tumor through its FC part. So the so-called antibody-dependent cellular cytotoxicity is induced. Essentially, it labels the tumor cell. You could say a HER2 overexpressing tumor cell and then this cell is killed by the activity of the natural killer cell. Should be mentioned that HER2 is overexpressed on tumor cells, but it's not absent on other cells, so it's not free of side effects. And it also facilitates or induces HER2 endocytosis and then HER2 intracellular HER2 degradation. So multiple mechanisms of actions, which are probably also the reason why this antibody is such a successful treatment, and its biosimilar successors because it's off patent now. And I think Trastuzumab is one of the most active areas of biosimilar production right now. So analysis of Trastuzumab, the old paper from Reed Harris from Genentech, our colleague I've been known for many years, for 30 years or so, who was involved in the development of Trastuzumab. And one of his papers that he published at the time showed the charge heterogeneity of Trastuzumab, which alludes back to, proteins are not homogeneous. So you see a major form, number 3 here. You see a second form, quite significant, number 1. And then you see a couple of other forms. 4 is still reasonably abundant, but 3, 2, A and B are relatively small. So this is a strong cation exchange profile with a salt gradient illusion, showing the product that is Trastuzumab as it is, before it goes into the patient. This just focusing on 3 sites, which is asparagine 30 in the light chain, 55 in the heavy chain, and aspartic acid 102 in the heavy chain, already shows that you have with these proteins, and that's the interest of analyzing metaprotein, you have a lot of combinatorial possibilities. This is only a few, but even only 3 modification sites can lead to a lot of different products because they, come in. You can have a deamidation at asparagine 30 and isomerization at asp 102, and all of these things can be combined. Here you see the percentages. So the main form is clearly most abundant. That's good. And that is the expected composition. But there's 14% already of a deamidation in one of the light chains, which makes it even more complex because you have everything twice in an antibody, unless it's a bispecific antibody, but usually you have 2 times the same site in an antibody. So this is quite abundant. And then here you have an isoaspartic acid, so an aspartic acid isomerization in 102. And these things can be, as I said, combined in different proteoforms, in different combinations. When it comes to biotransformation, that was a choice that we made. We focused on asparagine 55, so one of the 3 sites, and we wanted to know, is this an important site for activity, and is it actually modified in patients? When breast cancer patients receive Trastuzumab, is there an in vivo biotransformation at this site? And the reason is that if you look at this crystal structure, you see that asparagine 55 is in close interaction with this blue thing, that's a HER2 receptor, and this is the antibody, the one and the other, the light and the heavy and the light chain. So this was likely if this asparagine 55, we thought, is modified, then there is a reasonable possibility that it's a critical quality attribute and that this is important to prevent it. So just to give you an idea where these sites are located, so this asparagine 55 is in the heavy chain. It's followed by a glycine, and if you've read something about deamidation, you probably know that asparagine followed by a glycine is always a hotspot for deamidation because glycine is very small, and this allows the cyclic succinimide intermediate to be formed. I'll show it in a minute, and then to deamidation to proceed. As we are working with an LC-MS assay, SRM assay, we also choose a second peptide over here, and I can actually make them bigger because I have them here, to give us an idea of how much antibody is actually there. So we wanted to see 2 things, is the total amount of antibody changing, and relative to the total amount of antibody, how fast is the deamidation at asparagine 55 going. You can take this as an example because if you can design another signature peptide, you can also look, for example, at aspartic acid, asparagine 30 or aspartic acid 102. So this is, you look at particular sites. Here's a reaction mechanism. I worked on this many years ago, as you can see from this paper with my colleague Hanno Kolbe at Transgene in Strasbourg, and that was already at that time was well known. And it turned out to be a problem for one of our therapeutic proteins, so that's why we started to study it in detail. Here's the chemical mechanism. So if you have an asparagine in a peptide with this amide side chain, there is a possibility, especially if the next amino acid is small, like glycine, that this amide nitrogen makes a nucleophilic attack to this carbonyl carbon, leads to the elimination of ammonia and to this so-called imide or succinimide intermediate. This intermediate then can be hydrolyzed or is hydrolyzed either to the L-aspartic acid, so that means asparagine, L-asparagine is turned into L-aspartic acid, but it can also hydrolyze and open to the other side because this is pretty much a symmetrical molecule. And then you get L-isoaspartic acid. There's actually also, not shown in this slide, a possibility for that you can get D-aspartic acid through this mechanism, depending on whether the water attacks from the bottom or from the top of the ring. But let's keep that out of focus right now. So you can have these two different products actually from the deamidation reaction, makes it already more complicated. When Peter Püls was studying that during his PhD, our first concern was, and that is a concern I think you should always keep in mind, that we are not introducing artificial deamidation during our sample preparation, and notably during the trypsin digestion, which usually takes a couple of hours at elevated temperature. Trypsin digestion is usually done around pH 8. And when we looked at that, we can easily see that this peptide with the NG side goes down quite a lot, let's say, at pH 8. So we are inducing actually deamidation while we are doing the trypsin digestion. Here we see the other peptide, which gives us an idea how far we are in the digestion itself. So we want to have pretty much complete digestion without inducing too much deamidation. So this is a compromise that you have to look at for every site, because you can, it has to be experimentally evaluated. You cannot predict whether it's possible or not. There are now also lyc forms, I think, or trypsin forms that work at more acidic pH. So at that time was not available, but now for considerable amount of money, I must say, you can buy these enzymes that have also activity at acidic pH. Nevertheless, we found a compromise. So if we look at pH 7, there's little deamidation over, let's say, 6, 7, 8 hours, while we get fairly decent digestion. So we chose these conditions to do our trypsin digestion. So we look at the deamidation that's really happening in the patients and not the deamidation that we are introducing ourselves. Here are the LCMS analyses. I'm a big fan of chromatography and mass spectrometry, so I'd like to show you also the real data, let's say, and actually how nice they are, I would say, because within about 4.5 minute dilution time, so 10-minute runtime, you can measure the stable peptide, you can measure the non-deamidated peptide, you can measure the isoaspartic acid containing deamidated peptide, you can measure the aspartic acid deamidated peptide, and you can even see the steady state concentration of the intermediate succinimide. So it's a very comprehensive analysis that would be impossible with any other method that I know of, let's say. And this is all done in one chromatographic run of less than 10 minutes. So when we look at that, then we said, well, can we now, before going to patients, can we simulate this in vivo situation? So we essentially just put the protein in plasma at 37 degrees under sterile conditions, and we kept it for about 56 days. That's pretty long, but we took into consideration that breast cancer patients get an injection at about every 3 weeks, sometimes 4 weeks. And the half-life of an antibody is, in that area of multiple weeks. So it is realistic to look a little bit beyond, let's say, the 21 days. And what you can see in this graph is that the asparagine-containing peptide decreases. So there is deamidation going on under these conditions. The main product is the isoaspartic acid peptide, which is, or protein, I should say. It's just a surrogate for the protein, which is not very favorable because that really leads to a big structural change. We see a little bit of the aspartic acid peptide coming up, and we have a steady state, more or less constant concentration at a very low level of the succinimide intermediate. So this gave us the idea, okay, it's something that might happen also in patients, because, well, patients 37 degrees is also more or less in plasma. But before going to that, we also wanted to know, and we are still working on that, does it have an effect on receptor binding? So here you see 4 different traces from the same sample, and I'll go through them one by one. So the first thing you see, as it's down here, is the classical ELISA assay that everybody uses, let's say, the commercial ELISA, which is based on 2 anti-idiotypic antibodies, better to say 2 anti-idiotypic Fab fragments are used. So that means these antibodies recognize a site in the CDR region of Trastuzumab, and here another site in the CDR region of Trastuzumab. Actually, they are the same, so the same site. So in that way, you can make a sandwich, and then you can have an anti-FC reagent antibody with an enzyme coupled to it, and then you can generate the well-known colorimetric or whatever kind of signal in the ELISA. Before I explain that, I would like to show the other one. Let me go back one. That was not so clever. Let's see. So so this is the ELISA. I should have said that before, but the red dots is the stable peptide. So this essentially is the concentration in microgram per milliliter of Trastuzumab. And the first thing that struck us pretty much is that the huge difference between this concentration that we measure by LCMS and the concentration we measure by ELISA. So how can this be? So then we developed a HER2 receptor binding assay. So this is in an ELISA plate format, you could say. We, instead of coding a capturing antibody, we code HER2 on the surface, the extracellular domain. This one recognizes, Trastuzumab, well, as it should. And then we have an anti-FC antibody coupled to an enzyme to generate the signal. The big difference is here is we only need 1 intact binding site, and here we need both some binding sites intact. Otherwise, we cannot make a sandwich. And if we look at the results, the receptor assay, these yellow dots, essentially gives twice as much the concentration as the ELISA. So this indicates that these anti-idiotypic antibodies are affected by asparagine deamidation at position 55. Because if we look at the peptide, so here they are, if we look at the peptide that contains the deamidation site, it follows exactly, more or less exactly, the receptor binding assay in concentration. And this is the stable peptide, while this ELISA goes down twice as fast. So here, 1 intact binding site, and deamidation can be one on the heavy chain, one or the other. We don't know which one. They are identical, let's say. You can still get a signal from a ligand binding assay, but from an ELISA, a sandwich ELISA, you need 2 binding sites. And if you just measure a stable peptide, then you need no binding site at all. So you just measure the peptide. So this is interesting to see also from a bioanalytical point of view. You have the same sample and you measure it with 4 different assays, let's say, and you get 3 different results. So if you could say you measured with 3 different assays, the stable peptide assay, the deamidation sensitive LCMS assay, and the ELISA, and the receptor binding assay, then you get 3 different concentrations essentially. So it's very important to understand what's going on. And I think mass spectrometry is really instrumental in that respect, that it allows you to understand why these assays actually give different results. And then it's not a question of, oh, this is better or this is worse, but you know what you're talking about essentially. Otherwise, before this was the only assay that was available. So this was the pharmacokinetic curve that everybody was measuring. Now to patient, here's a breast cancer patient that's treated in the hospital, and these points are the injection points of Trastuzumab, so a fresh injection. And here we see actually in vivo biotransformation in the patient herself. breast cancer patient. So here we have the stable peptide, that's the concentration in microgram per milliliter. Here we have the non-deamidated peptide, which is a little bit lower, maybe 20% or so, 15 to 20% lower. And as you know, each time the patient gets a fresh non-deamidate injection, so this, you know, this deamidation of the antibody itself is actually faster, but it's compensated for that you give always a new fresh product. Here we see the ELISA, which again measures about 50% of what we have before. Here we see the isoasp peptide, which is, you know, not exactly the difference, but it's, you know, somewhere in that area of 20%. And here we see the aspartic acid peptide. So it kind of reflects of what we saw in the plasma incubation studies. So biotransformation is clearly important. And if I refer back to the receptor binding assay, it also shows that deamidation of asparagine 55 prevents interaction with HER2. So it also affects the activity of the product. So it is a critical quality attribute, which is okay in the product, but it is modified in patients. And we know now that it is modified at a different rate in different patients. We are now in a bigger clinical study together with the Dutch Cancer Institute in Amsterdam, and we are now studying many patients, and we see that not every patient shows the same profile. Okay, but beyond, I'm not sure all this thing is in there all the time, but maybe I can, no, I'll get it out there. But anyway, it's beyond asparagine 55 deamidation. Because we all know, we look at asparagine 55, but there are many other things going on. So how can we tackle this a little bit more? And this is an ongoing PhD project, actually 2 PhD projects. So we have plasma from the patients. We need to do an enrichment of our target molecule, which is Trastuzumab. We also work on Pertuzumab, both together. We need to separate the proteoforms. That's actually a very tricky business because we need to separate them at the protein level to keep the connectivity between the modifications. If we just digest everything together as we did before, then we would lose this. And then we need to analyze by, for example, LCMS, but also by functional assays like receptor binding or Fc gamma receptor binding for the antibody-dependent cellular cytotoxicity. Well, thanks to a long-term collaboration already with a company called Avacta Biosciences in the United Kingdom, we teamed up to make new affinity binders to do the enrichment. And these are based on so-called affimers. There's a lot of set between affimers, but this paper is a really great review about it. So if you're interested in it. So affimers are very small proteins of about 12,000 Dalton that can be engineered to bind different molecules. And so they have these 2 loops here. They're based on a protease inhibitor backbone. These 2 loops can be exchanged to various loops. And this is done at random by phage display. So Avacta has about a 10 to 10 number library of clones. And at random, these different loops are inserted, and then they are panned like a standard phage display approach to see which one binds to the target protein, which is in our case Trastuzumab or Pertuzumab. And we obviously do not want it to bind to other plasma proteins like albumin or IgG in general and so on. So there's a screen for Trastuzumab, for example, and a counter screen against other proteins. So very good. The other thing that's nice about these proteins, you can engineer them with tagging ligands. So you can, for example, there's no cysteine in the whole protein, so you can put a cysteine at the end for immobilization, or you can put a his tag in there for, you know, putting detecting moieties in it. So it offers a lot of possibilities, and they're very stable, more stable than antibodies. So in order to figure out, and that was done by Frank Klont in another project, a PhD student, so which of the affimers are suitable for our purpose, we used actually an type ELISA assay, but with a mass spectrometry detection. I think it's a very nice approach. So you can screen a lot of different samples of different affimers. So let's say you put the affimer on the plate. Frank still had an antibody, so this should be the affimer. You block the plate like you do in ELISA, you know, with a BSA and PBS solution or whatever. You do the capture of your, in this case, the green thing should be Trastuzumab. And that instead of putting a sandwich on to detect it, we essentially either do an on-plate digestion, as we do now, or we elute the protein from the plate. And this works actually very well. We've done this for many different proteins with Affimers, but also with antibodies. So essentially, you can do this like an ELISA. So everybody has the equipment in the lab for the sample preparation. But in the end, you get your protein out or your signature peptides, and you do an LC-MS assay. So when we do this, these are in unpublished data from Ola Dapo, PhD student in the group. Then we have here 16 anti-Trastuzumab Affimers, and you can see at 5 different concentrations. And then we prioritize them. For example, we take the top 4 dependent on the binding strength, and this is relative to a stable isotope labeled internal standard peptide. We have a counter screen, you could say, and these are 3 anti-Pertuzumab Affimers, and you can see as nice selectivity. They bind to Trastuzumab, but not to Pertuzumab. There's a plank with no Affimer, and then we have HER2 as a positive control on the plate. All this is done on 1 microtiter plate. And then later with on-plate digestion, you can just put the whole thing into the autosampler of the LC-MS system and analyze it. At the same time, so this is for enrichment, we're working on it also with Baubek Spanov, another PhD student. We developed the proteoform separation method. And we do this by so-called chromatofocusing. That was in collaboration with Alois Jungbauer in Vienna. How it works is essentially it's a strong cation exchange separation and dilution with a pH gradient. So here we have the starting material. This is clinical lot of Trastuzumab, which we get from the hospital. So there you have the inherent heterogeneity. You may remember a little bit, you know, the profile from Reed Harris. So it looks a little similar. Separation is better though. And then we do what we call stressing or incubation in PBS, pH 74 at 37 degrees. And then you see how this profile changes over 3 weeks. So 3 weeks is the time until the next injection of Trastuzumab in the patient. So you can see we have now an enrichment method. We have a separation method, but you can see there are many more forms than we can separate. So it's still a tricky business. But this is reality, let's say. That's what's happening. And now we are busy with looking at receptor binding, but also peptide mapping of the different fractions and the different forms, and also antibody-dependent cytotoxicity, which we do with colleagues at the National Transfusion Center in Amsterdam. Now we have an array-based assay, which works really well. Okay, let's conclude on that part and then get to the last part. So you could see Trastuzumab and its follow-up project, they have changed the way HER2 positive tumors are treated, that's for sure. At the product level, CQAs of Trastuzumab have been really maybe the most extensively studied CQAs, but in vivo biotransformation have remained largely unrecognized and unstudied. People were not interested in it, or there was also not the technology to do it, but they are clearly important. With modern sample preparation and bioanalytical methods, and mainly affinity enrichment and LC-MS, we can study these biotransformations, and we can hopefully relate these biotransformations to therapeutic response in the future, together with our colleagues from the Dutch Cancer Institute. Okay, so anti-drug antibodies, a short part. We worked on this a couple of years ago, and this was together with the Pompe Disease Center in Rotterdam. Pompe disease is a rare disease. It's an inborn error as a deficient enzyme, and the children have to receive this replacement therapy with alpha-glucosidase lifelong. So it's very important to measure level of anti-drug antibodies. They can interfere with activity. They can affect the in vivo half-life and obviously then reduce therapeutic efficacy. And there are different companies developing now drugs to down-regulate an anti-drug antibody response in these patients because there's no alternative to the treatment otherwise. So how do we do that? It's actually very simple. And I think that's a nice thing about these assays. They're very simple. So the commercial product is called Myozyme. And if you have anti-drug antibodies, then this myozyme will be bound to an antibody. So if you then use protein G beads in this case, you will capture all the antibodies in your plasma or serum, in our case was plasma. And you will also capture, once in a while, antibodies, anti-drug antibodies that contain myosin. So if you now use a signature peptide of myosin with LCMS, you can actually measure how much myosin was captured, which is an indirect measure of how many anti-drug antibodies were there. What is another big advantage of the LCMS method is that you can also follow the enrichment process. So you can just use a generic IgG1 specific peptide from the FC region. So you can see whether your IgG capture is working well. Oops, sorry for the background noise. And you can also see whether your depletion is working well. That means whether you removed human serum albumin by using an HSA-specific peptide. So the nice thing about these LCMS assays is that you can actually follow very nicely what is happening, whether your assay is working well. So here you see SRM traces of all these peptides. I don't need to go into detail, but they are nicely visible, let's say. So it's not that we're working at the limit of detection. And then you can make a quantitative accounting of everything. So let's say we start total, we have an ADA-free plasma, you know, that we just buy, let's say, a normal plasma. We have a patient plasma with a low ADA level, and we have a patient plasma with a high ADA level. So in the beginning, we set everything to 100%. So then we see what is left after we throw in the protein G beads. Well, if we have no ADAs, we have 85% left, and the other 15% or so come off in the wash. With the low ADA serum, there's a little bit less left, maybe 78% or so, and then some here. And the second wash is clean, so you can also follow that. And if you have a high ADA serum, or plasma, I should say, it's plasma, then yeah, nothing, almost nothing is in the supernatant because all the alpha glucosidase, the myosin, is bound to antibodies which are captured by the protein G beads. Now we should see that back. This is the free fraction when we elute. So when we elute, the picture is exactly the other way around. As expected, there's nothing coming off with the ADA-free plasma. The 10% that we are missing here is coming off with the low ADA plasma. And the 85% that we are missing there was actually bound to the beads via the anti-drug antibodies. And if we make the total accounting, this is quite nice. We get very good recovery, anywhere between 95 and 100%. So we can be sure that this assay works nicely, and it's also, many of our assays are fully validated according to the international guidelines. So here's a trace of that. So here we have the low ADA level. So here we have total, a peptide for the total glucosidase. And here we have the ADA-bound glucosidase, which is only 12% of that. In the high anti-drug antibody plasma, we see, 82.9% is actually bound of the total level. So it's really a very comprehensive assay. Kind of recommend it to, if you're looking into that, to try this out. This is also related to response to therapy. So this was together with colleagues from the Pompe Center at the hospital in Rotterdam. And here's a patient with reduced response. So has a high level of ADAs. So you can see that in the open symbol. So that's the level of ADA bound myosin. So it's, close to the total level. And here you have a patient with a normal or good response to the therapy, and there you have a very low anti-drug antibody level. So this is always important to make the link to, the actual, what does it mean for the patient in the end, what we're measuring. Okay, another intermediate conclusion, but it's very simple, I think. You can measure anti-drug antibodies quantitatively with LCMS in a very simple capture by a protein G affinity column. It allows you at the same time to follow the enrichment by an IgG1 specific peptide and the depletion of human serum albumin with an HSA specific peptide. Therapeutic response of the patients. Okay, so just a question. I'm almost at the end. So this section is not absolutely, let's say, necessary. It's interesting, but I could skip it if we would rather keep some time for questions. So maybe I should ask the organizer how to proceed.
We can proceed with the presentation.
Okay, so the couple more slides and not too many. I wanted to just allude briefly. I'm not sure my internet is okay. I just wanted to allude briefly to, much discussed point of, so what's in it if you use high resolution mass spec, and high resolution mass spec is Orbitrap or time of flight mass analyzers. And instead of doing an SRM assay where, select your precursor ion, you get fragments, and then you select certain fragment ions, quantifiers and qualifiers, which work very nicely. And that was the results I've shown. And we use a lot of triple quad instruments in the lab for quantitative bioanalysis. But you can also do PRM, where essentially you select the precursor ions, you do the same, but then you measure all the fragment ions at the same time. So you can a posteriori choose the fragment ions that you want on the computer, let's say, which is nice, if you don't, know, or you don't want to develop the assay before you say, well, let's get all the fragment ions and then we'll see which transition is the best. But this analysis has so far always suffered a little bit from sensitivity issues, but well, newer instruments, especially the newer Orbitraps, I think are getting close, but I have to honestly say, I don't think they match the modern latest generation triple quads in terms of sensitivity. So if you really need sensitivity, I think SRM in a triple quad is still okay, and they're getting very fast. So we have triple quads that can measure 100 peptides in one LC-MS run. So that's also pretty good. And that's usually enough if you know which protein you want to measure. But anyway, high resolution mass spectrometry. So we did some studies also with Peter Wilz again. Here we see a human growth hormone after tryptic digestion, and we asked a simple question. So what if, with PRM, so what if we change the extraction window? I'm not quite sure. Do you also see this thing? Well, anyway, now it's too late anyway. It's always blocking some of it. So, then what you can do when you have this acquired at high resolution, you can essentially adjust the extraction window and how exact you want to extract the fragment ion trace. So here you see we have the classical quadrupole, let's say, width of half a Dalton, half an atomic mass unit, and you can see you get a really fairly, wild chromatography trace with essentially no selectivity. If you go down to 0.07 Dalton, which you can do, this is on a TOF mass analyzer at resolution 30,000, then you can actually tickle out this peak out of the background noise, essentially remove background noise, other fragment ions that don't have exactly the same mass. So this is okay, and you can even go to 0.01 Dalton, but it doesn't really help very much. The thing is, if you go to very, very small extraction windows, which you can do on the computer, you really have to rely on an extremely stable mass calibration of your mass analyzer, because you can imagine if this peak should shift just a little bit to the left or to the right, it would wander out of your selection window and you wouldn't see it anymore. So you have to find a good compromise. But the nice thing about PRM, you can essentially play with this on the computer. You don't need to do extra analyses. You have all the data and you can just essentially extract whatever you want from this data. Another thing you can do is you can combine multiple fragment ion traces, which we also often do in the SRM. You can add them up essentially. Here's again the somatotropin, the human growth hormone. And here we have one fragment ion, two fragment ions, three fragment ions. You can see it doesn't really make a huge difference, but just to make the point that, you get nevertheless, more sensitivity from somewhere 75 counts to 120 counts. In other cases, it may add a lot more to your sensitivity. However, you can also see that you may get more background noise. So it's always a compromise between how much noise do I introduce and how is my signal increasing. So it's a signal to noise thing, as generally in analytical chemistry. But it's again a possibility that you can essentially just do on the computer. At the Interact, the big let's say thrust of high resolution MS is obviously not to digest anymore, but to work at the intact protein level. And well, the well-known challenges are there. We work with electrospray ionization, so we get a charged state envelope, as nicely shown here for the human growth hormone. And we have, isotopologues in each of these charge states. So it's a fairly complex thing. And then the question is, so what is then, which ion should I monitor? First of all, you lose a lot of sensitivity because you have to focus on 1 or 2 or 3 of these charge states. And you have to make a fairly broad window if you want to capture all the isotopologues, or you have to go in and select only a few isotopologues. And again, you lose sensitivity. That taken together with the lower ionization efficiency of proteins in general relative to peptides and the difficulties of chromatographic separation. The situation to date is that intact protein analysis in bioanalysis, at least, is not very frequently used and is not competitive in sensitivity with, let's say, the digestion-based approaches. However, I will show you at the end that for small proteins it is working, and there are fully validated assays we've developed and just published for insulin-like growth factor 1, for example, as a biomarker. But for bigger proteins like human growth hormone 22 kilodalton, or if I go on, the Fab2 fragments of Trastuzumab, or even the whole Trastuzumab, or you will see, at least at this level with this TOF analyzer, is the glycovariants in there, but not the isotopologues anymore. So there's not enough resolution. You would have to go to ultra high resolution. They're very expensive instruments. And also, I think you would lose a lot of sensitivity that way. So I don't see it yet. But, people are working on it, and especially with the top-down fragmentation, which we also work together with some colleagues in this EU project, you may be able to get information on protein modifications without having to digest the protein. Antibodies is nice that some people in Sweden some years ago found an enzyme called IDS, which allows you to cut at the hinge region of antibodies. Before that, it was done with papain, which also works, but not quite as selectively, but at a much lower price. Let's put it that way. Anyway, if you can afford to buy IDS, then it's a very nice enzyme with a very specific cleavage site. It's used a lot in the industry to cut the FAP region, so the antigen binding region, from the FC region. And if you also do a reduction in alkylation, like in this case, you actually end up with quite manageable fragments of 25 kilodalton. It's the light chain, the so-called FD fragment of the heavy chain, and the FC half fragment of the heavy chain. So then you're back in an area that you can manage. Here's a mass spectrum of such an analysis. So this is just one charge state. It's the +20 charge state of the FC half fragment. And here you can see at least, very easily the glycovariants, but also some other modifications. I believe there are oxidations in there and things like that. And if you can now do, and that's what we're trying to do, the top-down fragmentation, then you could localize these modifications without having to go further digestion, which is now called the middle-down approach, so to speak. So here it's after deconvolution. You can clearly see the various glycovariants. You can also see whether lysine has been cleaved off or not, and things like that. So let's say bigger mass differences down to, I would say, an oxidation is possible, but a deamidation will be very difficult to detect because it's only 1 Dalton mass difference. Well, I already alluded to the electrospray giving many charge states, and you would like to compress this charge state envelope, and it's not this is our work, but our idea was not, but people have worked on so-called supercharging. So to add agents like the metanitrobenzyl alcohol to increase the charge state of the ions generated in electrospray, and thus to bring the ions of large molecules down into an M/C region, mass to charge region, that is amenable to standard mass analyzers. And it also can enhance the sensitivity. So this is an agent that's been published a lot upon. We've done just a few tests so far. As you can see, it works. So this is without the additive, and this is with, but well, it's an additive. It's volatile, but somewhat not super volatile, let's say. So you also have to worry a bit about contaminating your mass spectrometer if you use it all the time, but it works. But you still have a fairly large charge state envelope. It's also the case that higher charged precursor ions fragment a little bit more easily because you simply get more collision energy into the ions, and that's one problem with big ions, that they don't fragment well because they have so many degrees of freedom to dissipate the collision energy. But if you give it a higher charge state, then obviously ions are accelerated more and you get harder collisions. This is just, I would say, preliminary data. We haven't really pursued it so far. Another way, and then I'm at the end, is to go the other way, to go to native mass spectrometry and to actually have lower charge states. Where lower charge state that helps, here's the the normal denatured Trastuzumab spectrum that we had before, and this is from the literature, a spectrum on the native conditions, and you can see you have here +53, and here you have the main is +24. So that means the resolution that you need to separate isotopologues becomes less demanding, still demanding, but less demanding. But on the other hand, you need mass analyzers that can go very high in mass to charge ratio. Most quadrupoles cannot do that, but TOF analyzers obviously have no problem with that. So there are some compromises, and you need conditions, ionization conditions that are native. So some buffer or so that is somewhat volatile. Ammonium acetate is very popular, ammonium formiate, but they are not completely, evaporating to nothing in your mass spectrometer. So also contamination can be a problem. And you need ion transmission conditions which work at a somewhat higher pressure, because you cannot have two forceful collisions in the ion transmission part and also in the in the interface, because otherwise you would essentially denature your protein again due to these collisions. Okay, it's that guy then, sorry. That was the assay which we developed. It's just Impress. You can see it. It's a front page featured material by Mark Bratt, a PhD student at the clinical chemistry department. And we actually developed an antibody-free protein bioanalysis assay at the intact protein level for insulin-like growth factor 1. I invite you pretty much to read it, but it's a selective precipitation procedure where first you precipitate all the bigger proteins, and then you take the supernatant, and then you add a more stringent precipitation agent, and then you precipitate your target protein, IGF-1. So I think for proteins below 10 kilodalton, definitely something to try, because this can be implemented in a clinical chemistry lab on a standard liquid handling robot, which they use all the time, because if you do drug analysis, you always precipitate your proteins. So it's pretty much the same procedure, just with different precipitation agents, and then you can recover your protein. And what is nice, you can actually measure it at intact protein level. So here we took IGF-1 and IGF-2 together, and we measured them actually in patient samples. We add a stable isotope labeled internal standard. We take the 8+ precursor ion here, which you saw probably on the left, and if you remember that spectrum. We do a CID, a collision-induced dissociation. It actually works surprisingly well, and we follow, kind of astronomic numbers, the B64 and the Y66, 6+ 7+ ion, and the B64 of the internal standard. So this is an SRM assay at the protein level. I think this was appreciated as a quite innovative approach, and it actually works. It's a fully validated assay, works in the clinical chemistry lab, and is now being developed as the reference assay for all IGF-1 assays analyses in the Netherlands, and hopefully beyond later on. Here are some traces, some examples from real samples. So IGF-1, IGF-2, the internal standard. Unfortunately, internal standards, the reference material, the quality of those is not always impeccable. As you can see here, you have two peaks. This is a disulfide bond variant, so it's a scrambled disulfide bond. So if you reduce it with DTT, it collapses into one peak. Well, that's what you get for quite a lot of money from the commerce as a reference material, as a qualified reference material. So this should actually be impeccable, but it is not in this case. And then we compared it with the standard assay, which is the hello. Yeah, I'm almost done. Sorry. So then you can see that actually the IDAS-cis, that's a ligand binding assay that everybody uses for IGF-1 minus the LC-MS, you have a positive bias. So you measure about 45% more with the ligand binding assay than with the LC-MS assay, which indicates that the ligand binding assay actually captures more than just IGF-1. We don't know what, but we can look at that. But the main point is that the IDAS-ICIS assay is not so reproducible in the sense that you cannot easily transfer it from one hospital to the other and so on. So that's why now a consortium in the Netherlands decided to use the LC-MS assay as the reference method, and then to calibrate the ligand binding assays, which are cheaper, obviously, based on this reference method. And so that to make an inter-hospital comparison possible, because otherwise it's difficult to diagnose. If a patient moves in the country or even beyond the country, then get different values at every hospital that you go. Okay, sorry for going a bit over time. The program I had seen, I was given 1.5 hours, but maybe that was my mistake. But now I'm done. So remember, no one size fits all quantitative protein assay. Always think about what is this assay really measuring. You never measure a protein. If you say I'm measuring let's say human growth hormone or Trastuzumab or so, always remember you only measure a very small part of the protein. Biotransformation, I think, will become more of a topic of investigation, and it may affect critical quality attributes, which are very carefully controlled when you make the product, but then they are kind of let go once the product is administered. Anti-drug antibodies can be monitored by LC-MS. I think it's a quite elegant way of doing this, and it allows you also to follow the quality of the assay itself. And yeah, high resolution MS definitely offers advantages, and as you know, it's developing very fast. But we will not. The instruments cannot overcome the principal limitations that electrospray generates multiply charged ions and then charged state envelope, and that we have isotopologues, which are hard to separate, so that our signal is distributed over many peaks, and this inadvertently reduces sensitivity. However, if we can get to more efficient fragmentation methods, as we showed for IGF-1, we can analyze IGF-1 at the low nanogram per milliliter level in plasma and serum using an SRM assay at the intact protein level. With that, I would just like to acknowledge some of the people I already mentioned: PhD student Peter Wills, Baubeck, Spanov, Ola Dabo, and Kees Bronsemer. My long-term technician in the lab, Natalia. Colleagues from the hospital, from the oncology department, Hilde and Jaurik, and Nico, long-term colleague from PRA Health Science, with whom we've been collaborating for more than 10 years in the area of quantitative bioanalysis, and also some of the funding agencies. And thank you very much for your attention and also for your patience, letting me go over time. So I hope there's still some time for questions.
Good morning, sir. A beautiful lecture by you. But sir, I have one basic doubt. Can I proceed with that?
Yeah. Sorry for the background
sir,
noise, but
no problem, sir. No problem. Sir, I do very well understood by your lecture. Yes, monoclonal antibody is very specific, but human biology is a very complicated system. Suppose we produce one monoclonal antibody, and suppose that antibody site on which that antibody is going to act is having a very more than 90% homology with another binding site, whether it is going to have a cross reactivity. That is my one question. And my second question to you is, sometime whether this cross reactivity is boon or is going to increase the output of monoclonal antibody. That's what, sir. Thank you so much, sir. Thank you so much.
Yeah, I'm not quite sure what you mean by output, but yeah, cross-reactivity is clearly an inherent problem of any affinity binder, I think, and it depends very much on the development of the antibody. So yeah, how you screen it and how you optimize it, let's say. I mean, Trastuzumab was based on a murine monoclonal antibody, the 4D5, and that region was then humanized, cloned into a human backbone. But I think there was, more than 10 or 15 years of research on 45 to evaluate it in all kinds of animal models and so on. So yeah, I don't think there's a general answer, but clearly, we know from experience that most antibodies shall have some cross-reactivity. So it can only be experimentally checked, what that cross-reactivity is. And then is the next question, is that cross-reactivity a problem in vivo, let's say, in the therapeutic application? Yeah, that is pretty hard to say. I don't think anybody can predict that really, but you can with mass spec, for example, put your antibody on a column or on beads and see, well, what is it binding? What kind of proteins, like a mini proteomics analysis. And then you can go through the literature and say, well, do we expect any kind of problems, with this cross-reactivity? And then it's also a dosing thing, because, well, you would expect that it reacts or it interacts much stronger with your target than with the, non-specific binding protein. So maybe in the end, it's not a problem. But yeah, I think it's very difficult. It goes into pharmacodynamics, so how much of the target is there, because, well, if you see how much antibody is injected, these are huge amounts, essentially. You reach 100, 200 microgram per milliliter of trastuzumab in a patient. You can imagine how much that is. I don't know how much HER2 is really there. So there's probably a very small percentage of trastuzumab that's really binding to HER2 on tumor cells. And what is the rest doing, you might ask? Yeah, that's a very valid question. And I don't know of any literature that people have actually investigated that. People really look at side effects based on target-specific side effects mainly. And that is in trastuzumab is usually HER2 on heart muscle and heart cells, cardiomyocytes. So that's the main side effect. That is kind of a dose-limiting side effect with trastuzumab, that some people get problems with the heart. But yeah, I'm only. Yeah, go ahead.
Have you observed any time sometime the cross reactivity is a boon? Instead of that, it really gave a very better result. Any kind of example it is there, sir, in the
cross-reactivity, out of the top of my head, I don't know, at least not for trastuzumab, but I know for many antibodies that are used for ELISA assays or so that they have cross-reactivity. You also see that often on Western plots. If you use a monoclonal antibody, you can see bands of other proteins, let's say, that should not react with this antibody. So I think, it's protein-protein interaction. How specific can you make protein-protein interaction if you're offering maybe 1 million binders to this antibody? It's asking quite a lot to say, oh, you should only bind to one. I think there's physical chemistry is the bottom line. There's hydrophobic interactions, there's hydrogen bonding, there's ionic interactions, and these have to be arranged in a certain way. But I don't think we can get, much further than what is possible in nature. And in nature also, antibodies are not monospecific usually. That's what I would conclude myself.
Thank you so much, sir. Thank you so much.
So I think it's we often cannot get much better than what nature does. Sometimes we do, and we can maybe it more efficiently, but where we have the 20 amino acids that are arranged in 3D structures, and they interact with another protein that also has 20 amino acids in another 3D structure, and this has to fit to each other. But if a binding site is very similar, for example, then it's, there can be cross-reactivity. I'm very happy with the Affimers because they are based on this huge clone library of 10 to the 10 or more. You can screen extensively, but you have to ask the question. For here, we ask the question, it should bind trastuzumab, it should not bind pertuzumab. We have also anti-pertuzumab, which should do the other thing around. It should not bind, obviously, plasma IgG, because otherwise we get all the IgGs. It should not bind major plasma proteins like albumin. So that's what we screened for. But we didn't screen for the other 900,000 proteins that are in plasma. So if you want to do that, then you have to go the other way around and maybe put then the affinity binder that you selected on the beads and see what does it bind. we did that for some of the ephemeras because we do a lot of proteomics, and we said, well, just put them on some beads, incubate them with plasma, do our washing procedure and see what's bound, and we still find 250 proteins bound, which I would expect, you know. So, but at a lower level, you know, on the gel, you hardly see them, but they are there. So this is never 100%, but maybe you can say it's 99% and 1% of other proteins bind as well. That's, I think, is a fact of life. I don't think you will get much better than that. Any other questions?
Thank you, sir. I believe there are no more questions in the chat box that I can see.
Oh, okay. Well, I hope it was clear and it was also interesting. Yeah.
Yes, sir. Thank you for such an insightful presentation on this quantitative bioanalysis.
Okay.
We really enjoyed it.
You're welcome. Thank you very much. And yeah, if there are still some questions, you know, you have my
yes.
You have my
so
email
I would
and
request
thing.
the participants, if there are any questions, you can write it in the chat box. We can wait for a minute, or you can even write it to Professor Bischoff. He has already shared his email ID on the first slide.
Yeah, don't be shy. It's always difficult online. I know that with my students too. Somebody has to start, otherwise nobody dares to write anything.
So I believe there are no more questions in the chat box, sir. Once
Okay.
again, I would like to thank you. Thank you for your time and for accepting this invitation to present. We're really
thank you very
grateful to
much.
you, and we hope that we stay connected in the areas of similar interest in future.
Yeah, I hope so too. That a real visit will be possible again in the future. Bye then.
Welcome.
All the best.
Yeah, thank you, sir. Have a good day.